folks, welcome. We're just going to give folks a couple minutes here to get logged in. Appreciate your patience with the technology.
University of Florida, and I'm co-hosting this with Bill Walton of Auburn University and Brian Callum of Louisiana State University. So why are we holding this workshop? Growers in the Gulf of Mexico have expressed concerns about very sudden, high levels of mortality in their crop, often in the spring and early summer, usually in adult oysters. These mortalities have not had clear explanations, unlike some events where the causes have been clear, such as lethal freshwater flooding seen last year in Mississippi and Louisiana, and of course, hurricane impacts. Now, back in January this year, Bill and I attended a grower organized meeting in Crawfordville, Florida, about high levels of mortality seen in the recent past in the area of farms there. Growers shared their observations and a number of possible explanations were discussed. In response, we suggested increasing environmental monitoring and doing some experimental field work to test various explanations to see if ways could be found to understand and to reduce or even avoid these mortalities. Then this spring, just a few months shortly after that meeting, growers in Alligator Harbor again saw devastating levels of mortality that could not be easily explained in both diploids and triploids. Pathological work was conducted by Dr. Ryan Carnegie at the Virginia Institute of Marine Sciences, and he found no evidence of disease, but instead the typical signs of spring-summer mortality. This again left us and growers asking why these mortalities happen. So I'll now turn it over to Bill Walton.
Thank you, Leslie. So in response to all these questions and concerns about whether these mortalities were related to PLOIDY, we decided a workshop would be a useful way to go forward so we could share what is already known about these mortalities and the effect of PLOIDY, let growers know what is being done or is planning to be done to get better answers, and obviously seek industry input about what's being observed and your priorities for future work. We all agreed that this would be best done in an in-person workshop, but we also recognize that this issue is so important to the industry that we shouldn't wait until large in-person meetings are occurring again. So here we are with a virtual workshop. We all know this is not ideal. Not everyone is tech savvy. It's not an ideal way to listen to industry input, but it is, for now at least, the best we can do, and we ask that you bear with us. Our hope is that we can follow up with growers in person as restrictions are lifted, and also this workshop is being recorded and a link to the video will be provided soon. So today, we're going to take a look at this panel of researchers that we have with us. We appreciate them taking their time. We've asked them to briefly explain their work. Our first panel will cover what do we know about oyster mortalities. In addition to Leslie and myself, we have Dr. Jerome Lepere of LSU. He'll talk about what we know about the effect of broodstock and different salinity regimes. Joseph Matt, or Joey Matt of VIMS, will talk about what he has seen in Chesapeake Bay, where they have observed a similar occurrence, a similar phenomenon. And we'll conclude that first panel with Dr. Ryan Carnegie, who will provide a real broader overview of the mortalities beyond PLOIDY. At that point, we will open up to some questions and answers. And I'll just actually skip ahead just while we do this. This will be our second panel, and I'll introduce those folks later. But a little housekeeping before we get started. We have asked speakers to keep it short and sweet. We know some people have expressed concerns that this is a workshop where people will be being talked at rather than heard. We want you to hear what is known though, but also get your questions and comments. So anybody that's done a video meeting like this, it is hard to have it work when we have even 10 to 12 people on the call, and I guess we're up to almost 100 participants right now. If you have any questions during the presentation, please, there's an icon to raise your hand that you should see, or you can type your question into the chat box, all right? Rusty and Natalie will read out questions during the question and answer session. And then, as I noted earlier for folks who came on, we've muted everybody. That's just ultimately somebody leaves their microphone on and there's chatter in the background. So that's just to keep the meeting manageable and effective for everyone. So we're gonna do our best to answer as many questions as we can before we move on to our next panel. I'm gonna go ahead with, we'll have contact information as well. I've mentioned the session's being recorded, And I guess with that, actually, I'm going to go back to Leslie, and you wanted to share your screen, so I'll let you start with our first presentation.
Thanks, Bill. So if you want to start with the first slide. So off-bottom oyster aquaculture is relatively new in the state of Florida. Actually, with the first leases issued in 2015, the industry has now grown to over 125 farms statewide. Despite the use of triploidy in oyster culture in other states, there was a need to understand the application in Florida. So in 2016, we were able to initiate a large-scale demonstration and evaluation of this oyster breeding process to our local conditions in Florida by working with our new growers. Basically, twofold objectives: document production, health, quality, and quantify the effects of culture methods and seasonal harvest. To do so, we provided 25,000 each of diploid and triploid seeds to growers over 2 occasions. The first set of trials occurring beginning in July and August of 2016, going 8 months and ending in spring of the following year. The second set of trials, again, similar number of oysters, both dips and trips provided in spring of 2017. A little bit of shorter grow out period, but terminating in October and November of the same year. The initial seed size about 25 millimeters or 1 inch. And we refer to both sets of trials as winter or summer. Bill said no figures, but here we go real quick. A lot of measurements we did, but I'm just going to highlight the 2 critical key variables that growers might be interested in. In this case, this would be shell height or length, and that's the measurement that you use to dictate your harvest size. So to orientate you real quick, on the bottom we have where the farms were located, the gear used on those farms. In this bar graph on the left is the value of the shell height. And to orientate you, there's your 3-inch mark there. So you can see that growth reached or exceeded market size in all of the locations here. The orange bars represent the average shell height for triploids, the blue for diploids. And you can see the trend here is that with the exception of 1 location, triploids far exceeded the diploids, probably providing a shorter crop time of 1 to 2 months. The other key metric that growers would be interested in is survival. And here's our winter trials, again, with average water temperatures per farm site running from 71 to 81 degrees. Exceptional high survival. This really was just a representative 3 bags that we did live dead counts. But more important to observe here is there was no differences in survival based on location, gear type, or ploidy type. Moving on to what we refer to as the summer trials. Again, farm on the bottom, gear type on the bottom, the same values of shell height on the left-hand scale there. There's our 3-inch reference mark. Obviously lower growth during this particular grow out period, but the same trends in terms of the application of triploidy being higher in all but 1 location. Survival over this period was lower overall. We did see average water temperatures in the various farms running at between 80 and 82 degrees Fahrenheit. The survival, really, there's no influence of triploidy. In fact, we just kind of saw the same in 2 locations, triploidy higher in the Cedar Key site here and lower, reverse higher in the Alligator Harbor site. Well, no really distinct trends. Now, there was 1 other grower in Alligator Harbor that had floating bags. Now, he started experiencing loss of his oyster crops in the spring and continued to the summer, and the project oysters were also affected. When we came back in November, there was 100% mortality of the diploid oysters, and we estimated about 20% survival of the triploid oysters from the project. Now, this was the first really large-scale mortality event in this particular lease area. It has occurred again in the spring of 2019 and just this past spring, both affecting diploids and triploids at that site. The pathology reports conducted by Ryan Carnegie at FIMS and Susan Larimore at FAU indicate no MSX, dermo, or pathogens of concern. Other observations are reflective of dying or stressed oysters. Very important to note that there was gonadal development in both the diploids and triploids. I'll let somebody else explain that one, as well as ripe males. Again, some key observations here. And if you look on the pictures here, you'll see what I'm referring to on the posterior end of these oysters. Both the mantle and gill abnormalities were associated with colonization of the shell in which the oyster is trying to ward off something. Also in the lower picture, you see discoloration of the shell caused by extensive deposits of an organic protein matrix mixing in with the calcium carbonate in the shell. On these events, we're also seeing increasing large blooms of amphipods, small shrimp-like organisms, and high siltation in the gear and on the oysters itself. This certainly fits the model for summer mortality, and in the case of Florida, we're going to have to rename this to spring mortality. I just want to say there's many more aspects of this study I just kind of briefly introduced, and they can be seen at this website here. All right, Bill, back to you.
Okay. All right. Let's see. We'll continue. All right. PowerPoint, share. Let me go to. And click through that. All right. Pull mine down. There we go. All right. You all should see my screen up there now. All right, I'll stay fairly short here. So this is what we know. This is Auburn University Shellfish Lab. What really prompted this for us was in 2016, we had an unusual mortality event. Farmers at several locations in Alabama, this is Mobile Bay here, the pins show where farms are generally clustered around the bay. And across several of those sites, farmers reported very high mortalities in 2016, and there were a number of causes suggested about what might be causing that. But there was this sense that triploids were dying at a higher rate. The problem with that was some farmers didn't have diploids to compare it to, and those who had diploids often had diploids and triploids of different sizes. So, I know this is maddening, but one of the first things we really wanted to find out was there really a difference? And so Pandora Wadsworth led this study. Without getting into really ton of details about the methods, essentially we put out paired diploid and triploid seed at 4 commercial sites in November of 2016 and left them out through October of 2017. And in those 4 sites, which are the 4 graphs on the right, the dotted lines are triploids. And so the take-home message is that triploid mortality was higher than their diploids by the end of the study at every site. What was interesting about that, though, was it was not a consistent amount higher, and the mortalities occurred at different times at different sites. So, for example, that first graph, Mobile Bay, that mortality is associated with a very low salinity event. And so you can see actually both triploids and diploids suffered from that, although triploids seem to suffer from it first. Oops. So that was the first study. That then led to Sarah Bodenstein's study of, are triploids more vulnerable to farm practices? And this is in revision. Take this with a grain of salt right now. We're working on the statistics of this, but Sarah went and deployed paired diploids and triploids at 1 site each in Alabama, Mississippi, and Louisiana at sites where people grow oysters commercially. The oysters were subjected to drying 0, 18, 24, or 48 hours and or grading and tumbling. And the idea here was we wanted to subject these oysters to farm stresses. There was some word from Australia that the growers tend to baby their triploids through the summer. And so we wanted to see if things that farmers were doing was that increasing mortality. So with those stresses imposed in May and July, we assessed later that summer, and I believe this is in August. The triploids, the first one was across all 3 sites, triploids did have higher mortality. It wasn't outrageous. It wasn't super high mortality, but it was statistically higher than the diploids. And what we see is these dark bars, the diploids are on top. And so there was pretty high mortality when they were dried for 48 hours and tumbled. But you can see, actually, we got substantial levels of mortality in triploids. Not only was it higher, but it also occurred when we dried oysters for 48 hours and didn't tumble them. And we didn't see that same elevation in diploids. So there does seem to be some exposure to the stress of farming, at least at the extremes here. 48 hours is longer than we would recommend anyone dry oysters in the summer, but we did see a difference at least at that level. I'll let Jerome talk about this, but we are also asking this question. This is in progress with Jerome, but does origin of the broodstock affect ploidy performance? And so I'll let Jerome discuss it, but we are looking at 3 sites in Alabama currently and looking at whether we see differences in different salinity regimes. So there's our website. And with that, I will go on to Jerome. And Jerome, I was going to share your slides for you. So let me pull that up. You'll have to unmute yourself, Jerome.
It's done.
There you go.
All right, thank you. And good afternoon, and thanks, Bill, for inviting me to present at this workshop. First thing I'd like to do is to tell you a little bit more about the results of studies we've recently conducted to try to identify and better understand factors responsible for mortality in oysters cultured off bottom. And these studies were conducted mostly with diploid oysters before mortalities in triple oysters became an issue. Some of those factors, like dermo, the disease caused by the parasite Percussus marinus, or very low salinity waters, have been known to be associated with oyster mortality in the Gulf estuaries for a long time, while we know a lot less about the occurrence and potential impact of other adverse environmental conditions such as hypoxia and high temperature or any of these factors in combination. Can we go to the slide 1? That's your last slide right there. Thank you. So what we have done in many of our studies has been to spawn wild broodstock collected from various estuaries along the Gulf of Mexico, and then determine how the progeny performed in field and laboratory studies and how long it took them to die when exposed to adverse conditions. Our main take-home message is that the progeny performance in terms of disease resistance to dermo or their tolerance to low salinity, hypoxia, high temperature, really is dependent on the broodstock selection. And I want to acknowledge Louisiana Sea Grant and NSF for funding parts of this work, as well as collaborators Brian Callum, Bill Walton, and Scott Rickard that were responsible for all the hatchery work related to those projects. Next slide, please. Now, dermo disease is well known to kill older, generally greater than a year old oysters in summer and fall when conditions are most favorable for the proliferation of the parasites. Mortality typically begins when parasite number exceeds about 500,000 parasites per gram of oyster tissue. And progression of the disease or how fast it kills the progeny will depend on what broodstock is being used. As you see in the top figure, which shows differences in how fast the number of parasites increases, and in the bottom figure, the associated mortality between 2 oyster stocks, the progeny of broodstock collected from 2 Louisiana estuaries. Can I have the next slide, please? Very low salinity, less than 4, is also well known to cause oyster mortality, especially when water temperature are greater than 80 degrees Fahrenheit. Exposure to high salinity can also cause significant mortality, but this condition is not frequently found in Gulf estuaries, except maybe in hypersaline lagoons in southwestern Texas. From field and lab studies, we have found that stock derived from high salinity waters, such as from Parker and Sharon in Texas, fare the worst at extreme low salinity, as you can see by the stock high mortality at the salinity of 2 ppt in the top figure. In contrast, stock that derive from relatively low salinity estuaries, such as those estuaries in Louisiana, fare the worst after acclimation to extreme high salinity, as you can see by their higher mortality in salinities of 38 and 44 in the bottom figure. And the arrows indicate when target salinities were reached. And it's again clear that the tolerance to extreme salinity depends on what broodstock they came from. Next slide, please. Hypoxia is another factor of concern, but we really don't know much about how frequently these conditions occurs in Gulf of Mexico estuaries, or how much hypoxia you have in a culture, bottom culture situations. We have recently exposed the progeny of oysters from several wild broodstock, and I found that there are significant difference in how long they can tolerate hypoxia and survive with their shell closed, as you can see from the two top figures. Now, the arrow indicates when the hypoxic conditions were reached, and the bottom figures are just control aerated water where was no mortality. So again, broodstock origin has significant impact on the tolerance of the diploid progeny to hypoxic water. Okay, next slide, please. Now, high temperature is another factor that has been poorly studied, but that could cause serious mortalities, especially when combined with other adverse factors, such as extremely low salinity. For example, oyster acclimated to salinities of 4 and 38 died much faster than at salinity 20 when the temperature was raised to 96 degrees Fahrenheit. And the arrows indicate when those target temperature was reached. As with the other results, we've shown again that broodstock selection has a significant impact on the progeny tolerance to high temperature. And high temperature is something we're going to have to worry about considering the global warming. If you have the last slide, please. All right, now, because broodstock selection can be such a determining factor in the survival of diploid progeny when exposed to adverse conditions, and because of the reported unexplained mortalities in triploids, oysters, when we submitted a proposal in 2018 to NOAA Aquaculture Program to look at the effects of the selection of diploid broodstock when crossed with variable tetraploid has on the performance of the triploid progeny. And the grant was funded last year, and free stock of diploid and triploid oysters were produced from wild broodstock collected from Louisiana estuaries and are currently being grown in Louisiana and Alabama. We are just beginning to see some unexplained mortality in those triploid oysters, with mortality right now at less than 10% at the end of April. Using those oysters, we're also trying to determine the underlying physiological and molecular reasons why the triploid oysters are dying while diploid oysters are not. Additional funding was just obtained from Louisiana Sea Grant to try to answer this question. Finally, we also be comparing the tolerance of those diploid and triploid to extreme conditions, such as high and low salinity, high temperature, and also be looking at resistance to dermo to determine whether the triploid oysters may be less tolerant to stress than diploid oysters. Now, this work would not be possible without the help of all our collaborators and graduate students, and I want to thank them all for their great work and effort. And thank you for your attention.
All right, thank you, Jerome. Appreciate that. Let me pull up Joey. Joey, actually, you're going to share your screen. Are you able to go ahead and do that?
Yes.
All right, you should have the ability to do that without, I'll stop my share. Perfect.
Great, thanks, Bill. Now for something a bit different, moving to the Chesapeake. Since at least 2012, there have been reports of unusual mortality events in the Chesapeake Bay. 2014 was an especially acute year for these mortalities, as the Virginia Institute of Marine Science received reports from 14 farms in Virginia. On most farms, these mortality episodes lasted about a month and was killing more than half of the affected stock. And the common criteria among these reports was the time of year, which was late spring, and the type of oysters that were affected, which were near market-sized triploid oysters. Since 2014, VIMS has received at least 8 reports from farmers in the Chesapeake Bay with matching criteria, although most have been less severe than the reports in 2014. It's also worth noting that VIMS got several reports just this past year. And what made these mortality events unusual is that they were not linked to the typical stressors, such as heavy infections of MSX or dermo, extreme temperatures or salinity, or sudden changes in salinities or temperatures. On the right here, we have a diagram of what an episode looks like, a sudden spike of mortality within a month's time in late spring, and then also a map with some regions where BIMS has received reports in red. There are plenty of questions following these mortalities, but I want to highlight those considered immediately testable through field experiments. The first relates to the effect of location. From reports, the relative importance of type of oysters being grown versus the location couldn't be worked out, so we wanted to know if you grew the same stocks at different sites, would mortality vary? The next question relates to the type of triploids being grown. We knew triploids could die, but we didn't know if a specific genetic type of triploid or triploid strain was responsible. And the third question was about ploidy. We knew triploids were dying, but because all of the affected farms were exclusively growing triploids, we didn't know the relative susceptibility of diploids. And last, although not in the reports, we wanted to find out if there were major changes in water conditions related to these mortalities. An initial institute-wide response looked into these questions, and the results of this investigation were published by Gavelou et al. In 2019. They found mortality varied by site. Multiple strains of triploids had a spike in mortality in late spring. They found mixed results on whether diploids were as susceptible as triploids, and found no evidence for major changes in water conditions prior to or during the mortality. For the rest of this short talk, I'd like to focus on a project that was a follow-up to Gavelou et al., which was Matt et al., the results of which were just recently published. Because it had the luxury of more time, it was a more controlled experiment than that in Gavelou et al., but it had many of the same testable questions. So I'm going to briefly walk you through the methods and results from that follow-up study. In 2015, 4 genetically different triploid groups, as well as a reference diploid group, were spawned. That June, the triploids and diploids were deployed to 4 commercial sites in Virginia, 1 on the western side of the bay and 3 on the eastern side of the bay. And when the oysters were about a year old and were nearing market size, we started closely monitoring mortality and water conditions. And we were expecting to see a spike in mortality in late spring between May and July, as this would have fit the criteria for previous reports. We saw the mortality event at 1 site, and we only saw the mortality event in the triploids, represented in black here. During the mortality event, mortality in the diploids, represented in white, was less than 5%. Also, there was no evidence of major changes in water conditions during this event. Here I'll point out salinity, which was between 17 and 19 parts per thousand during this time. So Matt et al. And Gavelou et al. Share many of the same conclusions, with the main difference being that in Matt et al., they found that diploids were not as susceptible as triploids. Finally, some take-home message of the earliest research of these unusual mortality events in the Chesapeake. We see that these mortality events vary by site and year, but the timing, late spring, is consistent. We found multiple strains of triploids are susceptible. There's evidence to suggest that diploids are less susceptible, which I think justifies referring to these mortality events in the Chesapeake as triploid mortality events. And last, we've seen that they can occur without evidence of stress from temperature, dissolved oxygen, or salinity, which I think may be especially important to consider when relating these mortality events here in the Chesapeake to those in the Gulf. Thank you.
Thank you, Joey. Ryan, we'll go on to you. Did you? I've forgotten, Ryan. Did you want me to share my screen, or did you want to share your slides? And you're on mute still.
Is it me?
There you go. It is your turn. Yes. Do you want me to share my screen for your slides, or did you want to do it? Can anyone hear Ryan?
Can't hear him. He's muted.
Ryan, we can't hear you. You're muted. I've got your screen up, though.
I should be unmuted at this point.
Okay.
Can you hear me?
Yes, and we've got your screen up. If you want, I can share mine, or you can switch over to yours.
Can you see my screen here, or have I shared?
You can not yet. Let me.
So why don't you just advance me,
okay.
Bill? We'll start on the first slide there.
Fantastic.
Okay, so so we've been working in this area of unusual mortality for several years. since these initial events in Virginia in 2013 or 14. Probably something on the order of 110, 120 samples have come through the lab to this point, coming from industry. Probably a third of those have been, a quarter to a third have been Gulf Coast samples. So we're forming sort of a pretty good impression of sort of what a standard sample would look like that comes through pathology. In addition to that, we've been working with, in the experimental context with the VIMS ABC, and some of our own experimentation and so on. So we've been forming some impressions about what is going on and what the questions are from this experience. And clearly our thinking, you know, sort of from where I sit in the pathology lab, aligns pretty well with what everyone's been saying to this point so far. I mean, clearly triploids are, you know, showing higher levels of mortality than diploids. This is mortality that tends to be focused in the springtime and early summer. We have this perception that it's associated with with gametogenesis and the annual sort of peak in reproduction. So probably some metabolic stress, as if the animals are sort of, you know, running close to the edge in producing gonad, and triploids are producing gonad. You know, even if they're not producing a lot of mature gametes, they're still producing a mass of gonad. And all of this is happening at a time of year when the food environment is not ideal. After the passage of the spring bloom, this is a time of reduced food quality and quantity, only increasing the stress on these animals at a time when the water temperatures are high. So the metabolic demands are high. So there's a picture that's definitely taking shape, but that can make it sound like it's all well resolved when we still have a ton of questions that we have yet to ask. And Bill, if you could just go to the next slide, please. So first, you know, we know that triploids are dying at higher levels than diploids, but diploids are dying too. I mean, in some cases they're not dying, you know, perceptibly. In some cases, in a fair amount of our data, the published Gavelous study, that's trailing along triploid peak in mortality. You know, we've been sort of making the case, and we made this argument, you know, down in Wilmington, and I'm sure many of you heard that, you know, a couple months ago, that, you know, while we need to focus on triploids and what triploids are doing in particular that might be leaving them more susceptible to a health challenge, there is the question of whether, like Jerome was saying, you know, are there other questions that we might be asking? Is it something in the provenance of the selectively bred lines? You know, where the broodstock is from. I've raised questions about whether levels of inbreeding in selectively bred lines might have something to do with it. Is there something inherent in our collection of broodstock, where it's coming from, and its passage down this domestication pathway that's leaving these animals more susceptible? These are questions that I still think we need an answer to. Bill, if you could go to the next slide, please. So the pathology that we see raises questions about whether or not there could be environmental stressors that are contributing to this dynamic. And this is a section of histology from an early event. What we see at the top of the image is essentially normal gill structure. And at the bottom, you can see basically this frayed appearance of the gill, where it's been affected by something, and it's displaying some level of erosion. This is a sign that we see fairly often associated with these mortality events. And we don't see anything in the way of obvious micropathogens, dermo or MSX in the Atlantic, or anything else in terms of microbiology or protozoan parasitism that could explain these signs. So, you know, we continue to be drawn to questions about what environmental stressors could be triggering these pathological signs to these organs that are exposed to the surrounding seawater, most prominently gills and mantle. And you know, Jerome mentioned hypoxia, and that's something that's always coming to our minds about potential stressors as animals are you know, crowded in cages, undergoing diurnal low oxygen challenges in the wee hours of every morning. Is this something that could be environmentally tipping these animals close to the edge? But we don't know exactly what these stressors are. Hypoxia is one possibility. There could certainly be others in these different environments across which this mortality is being displayed. Bill, if you could show the next slide. And this is a slide, so Leslie showed this image earlier, and she reminded me of this yesterday, and I pulled this into my presentation. We also have these questions about whether there are and what we can see, if you look at the bottom center oyster, the top right oyster, you can see these little trails of brown that are leading from the edge of the shell. And these are the areas in which the mantle and the gills are sort of retracting or being recessed a bit by some interaction with whatever that is that's colonizing the shells from the outside. This suggests some sort of biological challenge that is stressing these oysters that has not yet been identified. So this is another question, whether or not there's something microbiological that is at work that's contributing to this pathology. And then just the last slide, Bill, please. So, I mean, we're thinking about how best to sort of continue to dive in and pursue this. And we have obvious questions, I think, that can be addressed at relatively small scales with experimentation on different farms and research farms and so on. The microbiology of these systems as they travel through these annual challenges would be one obvious question. And there are a whole lot of other ones that we could address at the small scale through experimentation. But I think what we really need to do at the same time is go much bigger in terms of trying to understand what's going on at really the scale at which this is happening from, Atlantic Canada all the way to the Gulf, looking at what are potential commonalities in terms of the oysters that are being grown, whether they're diploid or triploid, how long they've been in domestication, what is the mode of culture, are they floating on the surface, are they on the bottom in cages, what kind of systems are they being grown in, are these small, very small tributaries of larger estuaries that don't flush well, are they big open systems, and so on, to try to put all this information together to figure out, all right, what really might be commonalities across the challenges, across the vast geography at which this is being observed. So I think we need to move forward thinking both in terms of small scale, taking this apart, and large scale, who is having the problems and what are the similarities that we might raise that we can go after in terms of mitigation. And I'll leave it at that.
Hey, great. Thank you to the first round of presenters. With that, we'll go ahead and take a minute here for some questions. And just as a reminder, you'll need to type your questions into the chat box. And we'll see if we have any questions.
All right, I have a few questions actually pertaining to gill abnormalities. If you guys could elaborate a little bit more on that. And then also one of the questions was, can you see the gill stressors from the visibly from the naked eye as well?
So perhaps I should handle that one, Bill
Yes.
and Leslie.
Yeah,
Yes.
I'd say so, and maybe
the
Jerome may have
stressor-
something to add, but
yeah.
let's start with you, Ryan.
Yes, certainly in our experience, most of what we see in terms of the gill pathology is not something that would be obvious macroscopically, like to the naked eye. It's something that's only going to be apparent when we look microscopically at what's going on. The slide that image that both Leslie and I showed of those oysters with the really exaggerated retraction of the gill and mantle that's been unusual in our experience. So generally, you'd need a microscopic analysis to resolve that.
All right, we also have a question from Allison saying that someone mentioned that there was a spike in amphipod populations during these times. It wasn't very clear that it was happening throughout the whole region of increased mortality, but could these be competitive parasite relationships between the amphipods and the oysters?
I will first just say that these are observations from growers in Alligator Harbor and in Oyster Bay an adjoining water body. I think they've been observing increasing populations over the past 2 to 3 years now. I would think I will go back to Ryan on the nature of that interaction with the oysters themselves.
Yeah, so the amphipods, I've been, communicating with Leslie and Bill, and some of the growers about that. And, one of the questions has been, we don't know what the MSX intermediate host is. Is it possible that these amphipods are intermediate hosts and contributing to one of these MSX or another protozoan parasitic disease that might be affecting these oysters? And we see no evidence of that whatsoever. My own, hunch is that the amphipods, are, maybe they're more than just this, but they're, I think, maybe more symptomatic of larger problems that are happening sort of within the microenvironment of these bags and cages and so on. If the conditions are such that, maybe flow isn't great and there's a certain siltiness that's developing, fine sediment is accumulating, that might be promoting the colonization by some of these invertebrates that are associating there. So I'm sort of viewing it in that way, but I don't have a lot of firsthand knowledge to base that on, just sort of the reports I'm hearing.
I'll jump in, and it's interesting. I don't know if the folks over in Louisiana have seen something different in their oysters, but we have typically not seen that on oysters in Alabama and Mississippi when we've seen those mortality events. So it may be causing it. It may be related to some of the mortalities that we've seen. It may be associated with something else that's going on with the oysters too. That's one thing we've got to figure out for sure. But we have seen oyster mortalities without those present.
Yep.
We also have some questions about the shell deformities. Are we seeing any type of parasitism from Polydora? And has anyone done any x-rays of the shells, Ryan, that you showed earlier with the brown trails on the edges?
I think I'll jump in there. The picture of the shells that I showed, actually that was just shared with Ryan and I think Bill just a few weeks ago. There's been no follow-up or looking x-ray, but I know that Ryan indicated that that's certainly showing a deposition of organic matter, which could be associated with this siltation. Again, those shells were from Alligator Harbor. That it could be associated with that excessive amount of siltation, organic matter being incorporated into the calcium carbonate matrix of the shell. Ryan, I don't know if you can add to that.
Yeah, the only thing I would add is that, I guess I would reiterate that yeah, there could be some level of response to something that's, sort of coming in from the edge there with some of those, and some of the images you've seen. Most of what we've seen in Gulf samples, these are generally very clean shells. They're not heavily colonized by shell epibionts at all. They're relatively thick shells, I think, by virtue of the tumbling and the husbandry that's going on down there. So they're not being penetrated to the interior by Polydora and boring sponges, such that the oysters are producing a lot of conchiolin on the inner surface of the valves. Like, they're nice and clean and white. And if that was not the case, if there was a lot of conchiolin being laid down, we might envision that would be stressful enough metabolically, producing all of that protein to wall off Polydora, that it could tip the animals over the edge. But that's not what we're generally seeing at all.
We also have a question saying, for the triploid seed that growers are getting from hatcheries in Florida, where did the tetraploid broodstock originally come from? And is this stock being rotated for genetic diversity, or is the sperm essentially from the same genetic stock?
Brian Callum's on the call. You want to talk to that, Brian?
Yeah, so the tetraploids that are used in the Gulf originated at LSU and have then They were developed at LSU. Some were transferred to Auburn and Alabama. More were created there, and then those have gone into Florida as well. So the tetraploids that are being used in recent years in Florida either came from Alabama or LSU, but all came from the same pool of genetics.
And I think in our second panel, we'll talk a little bit about the efforts to expand the tetraploid broodstock in terms of doing that. But I know there's always been an effort to, we certainly are aware of not wanting at Auburn to have that tetraploid broodstock narrowed down too much. So there has been an effort to bring in new genetics to that, but certainly working on some other lines, as we'll discuss later. And if I could just, by the way, one other thing, just to go back to that mudworm, you know, one of the concerns for growers that look at stuff like this, one of the issues is you might very well see, if you have a batch of oysters, that you might see mortality and you might see mud blisters, but that's also could be associated with the batch that you might not have been handling as much, might not have been doing as much husbandry with. And so, you know, this is why, you know, this is like, this is, if this were an easy problem, somebody on this panel here or one of you all would have figured it out by now. So I don't think this is an easy answer because it gets complicated. We could find a bunch of oysters with mud blisters and see high mortality, but that doesn't mean that those mud blisters cause that high mortality. They could be responding to whatever's going on in that basket or that bag. That's, in my experience, we've looked at a lot of mud blisters and we have not seen that associated with mortality. We don't even really see it affecting the condition index of the oyster all that much. So I'm sorry.
We have, for floating cages, are the oysters at all exposed to air during the spring and summer? Could this increased air temps have any impacts on the oysters?
Bill, you're up.
I will certainly, the air drying is happening probably for the adjustable longline system and the floating cages, at least it can. The floating bags is a little harder to get the oysters dry. Some growers are using floating bags where they stack them like this. They flip them, and so those bags may come out. But desiccation is actually one of the techniques that we have recommended at Auburn to control biofouling. That doesn't mean every grower uses it. We have some cautions on that in terms of avoiding the heat of the summer. So we would dry for 24 hours every week routinely, except once you get into 90-degree water temperatures and 90-degree air temperatures, we knock it back to about 12 to 18 hours and avoid the heat of the day.
For the brown edges that we saw on these shells, is this only seen in the suspended oysters, or is this also being recorded in bottom wild and or cultured oysters?
Well, I'll start to answer. So the samples that were sent to Ryan Carnegie from Alligator Harbor, the pictures that you saw, those were all from either, I think, floating bags or floating cages. There was no bottom culture oysters that were selected for this. I'm not even aware of any bottom cages being used at the Alligator Harbor lease site. That's probably the short answer. I don't know if Ryan has anything to add to that. So we don't know if it's necessarily gear related. It was all floating culture methods, off bottom methods.
Joey, what was your experience in Virginia? What did you see with your growers with the gear types?
Well, with the project that I was involved with, we used a common gear type at all the sites, which was bottom cages. And there was fouling on, oysters at every site, and the fouling varied a bit by each site, but I don't think there was evidence to suggest that the fouling had anything to do with the mortality event we saw, because we took such good care of those oysters.
Okay. Are we caught up on questions, Natalie?
We are. I have a few more questions, but I think they'll be answered by the next round of panelists.
Okay, with that,
Well, just before we do that, let me just offer, like, does anybody, like, I think we've got a few growers here. We've got just a minute here. Does anybody have any, were there any other growers that wanted to chime in about either with a question or a comment? All right, folks, a minute. All right. Certainly, if you do, certainly we'll provide some contact information. So please don't hesitate to follow up with folks here. Rusty, sorry about that. Go ahead.
No problem. And at this point, we'll move on to the next panel to discuss what's being done currently and what the plans are in the near future. And as Bill said, there'll be some additional time at the conclusion of the panel for questions. So as a reminder, just put those in the chat box.
All right, so you can all see the agenda for the second half here. So in this panel, we've got Tom Rossi of 4Cs is going to talk a little bit about the development of some new tetraploids, which came up. He's going to be followed by Dr. Huiping Yang of University of Florida, and she will talk about some of the development of Florida tetraploids and her cryopreservation work. Scott Reichard, I think, is going to come upstairs from the spawn that we're doing today. He's going to talk about some of the work being done at the Shellfish Lab. Dr. Kelly Lucas of the University of Southern Mississippi at the Gulf Coast Research Lab is going to talk about a regional consortium that she's leading up, working on a breeding program to develop improved diploid lines. And Dr. Brian Callum is going to talk about what they're doing at LSU. Then at that point, we'll jump back up to Chesapeake Bay, and Dr. Stan Allen will talk about what VIMS is doing with their program and a bigger picture, probably. We're going to wrap up with Leslie and I talking about some of the collaborative efforts to evaluate field performance of triploids from different broodstock and some of the other things we're trying to do. But then we'll go back to an open question and answer session before we wrap up at the top of the hour. So with that, I'm going to turn it, I'll stop sharing my screen and hand it over to Tom Rossi.
Okay, thanks, Bill. No slides from me. I just want to give a brief presentation here about what's going on presently. First of all, everyone knows about the problems in the Gulf, and you've heard that these problems, similar kinds of problems in triploids, are present up and down areas of the Atlantic coast. I can tell you that in our work, we've had relationships with people in most of the major growing areas of the world, and triploids tend to have special problems. As someone told me early on, they get the worst first. So what's happening in the Gulf may not give you any comfort, but there are other growers around the world that suffer similar kinds of problems. So our job in this process in the industry really is to facilitate the distribution of tetraploid oysters. It's a rather difficult process. You know that we have only had tetraploids available in the Gulf of Mexico for past 6, 7, 8 years maybe. And that's owing mainly to the difficulty in creating this animal. It takes multi-generations. There are difficulties in survivability, which I'm sure you'll hear from Huiping, who's been working on it for some time now. And you know, we work with industry partners to try to get as much diversity into the tetraploid population as possible. But as I think you also heard, the fact that you have a good trait as a diploid doesn't necessarily transfer into the tetraploid because of the process by which the tetraploids are created. So we have not figured out how to make a tetraploid parent that will give you a reliable triploid seed. We have really kind of taken to a little bit of trial and error, which is what we've been working on for the last couple years in Florida. We have Gulf-wide tetraploids that Brian mentioned. There are tetraploids growing in LSU now, and there are tetraploids growing at Auburn University. And we have, over the past couple years, worked on developing a line of tetraploids in the state of Florida. And this year is the first year that we've had the successful population to the extent that we have enough commercial viability. We've recently distributed tetraploids to all of the hatcheries that are producing seed that wanted to produce triploid seed. So this is the first year that you will be able to buy seed from hatcheries in the Gulf that the triploid seed is developed with the father side of the parentage from a tetraploid produced in Florida. So we've encouraged the hatcheries to provide this as an additional product. Again, it's going to be available. It's up to the growers to decide what they buy, but if you talk to your hatchery suppliers, you should be able to make that kind of choice. Would further encourage the growers that are involved here to track the results of the various seed products that they get from their hatcheries. The only way we're going to know whether we're heading down the right road is to get some results back from growers. Hopefully something that's going to come out of this is a more scientific study comparing various triploid seed grown from Florida tetraploids and Gulf-wide tetraploids. But as growers, you'll be able to see the results yourself, probably more so than any scientific event. So keep track and respond to your hatcheries. We'll be in touch with the hatcheries. We're sort of looking forward. No promises made here. Again, it's a trial and error. We don't know how these triploid seed are going to produce next spring when the typical mortalities usually occur, but we'll be looking for feedback from the growers, and so we know where to go in the future. The only other point I want to add is that we'll continue to develop additional lines. We'll use Florida stocks. We'll use stocks in different areas of the Gulf to develop additional tetraploids. But until someone figures out what the right formula is, we won't know what we're going to use as base stock. So as we get additional tetraploid lines developed, we'll let the hatcheries know and be able to make choices with them as to the kinds of seeds that can be produced. Thanks.
All right, great. Let me appreciate that, Tom. Let me pull up Huiping Yang's talk. Let's see, everyone should see that screen now. There you go. Huiping, you may be muted at this point. I can't hear Huiping.
She's muted.
Oh, there we go. Gotcha. Yep,
Okay.
absolutely.
Sorry.
Go ahead.
It's good that Tom talked about the tetraploid before me, and so after I came to joined the UF in 2014, I heard a lot about the tetraploid need for the Gulf oyster industry. Next one, please, Bill. So in 2017, we'll be able to get a fund from the Gulf States Marine Fisheries Commission. That's a 2-year fund, like a first year and the second year. But we will be able to work for 3 seasons in 2017. And that's the first year we start to work, get some chemically triploids, and for the 2018 and 2019, we are trying to do some produce some tetraploid founders by the difficult way. It's very difficult to do the 3N plus like a cross with the 2N. Currently, after this 2-year funding, we do not have funding anymore, but we do have a proposal. It's pending, and the goal based on the industry input, and especially through the Florida Shellfish Aquaculture Association, we heard that they would love to have establish a kind of like an extension program which help the industry to understand the triploid tetraploid technology. Next, please. That's the summary which I did. For this project, we did like 2017. We did got like wild oysters from Florida. We probably, I remember, is we got oysters from 7 locations, but only 3 locations the oysters spawned. We did I would say a lot of farms in the Florida, they helped to culture the oysters. You will know that for me, the lab, especially I'm kind of a new faculty member in the US, I have no capacity, there's no hatching, no grow out, nothing. So the industry helped a lot, at least some of the farms to help. Next, please. And then during this process, I know that the flow cytometer has been used for PLOIDY determination for like many years. Dr. Allen is in this group in today's seminar. But for us, my lab do have a flow cytometer. We'll be able to use this flow cytometer, build up the protocol, and use it for this product. And at the same time, we try to the protocol be able to help some other products. For example, Leslie talking about her product at the beginning, that we helped to do the PLOIDY determination. And also for Tom Ross's tetraploid product, we helped for that PLOIDY determination too. Next, please. So in the second year product, what we do is 2018, 2019 during the spawning season. This is kind of a group effort. A lot of graduate students, visiting scholars, everyone is helped with this work. We did be able to screen all of the chemically produced triploids. We found some females. About the female percentage from the triploid is about 1.57 percent. That means 100. You get like 100 triploids, you probably find one or 1.5 triploid females. We did 2018. There's probably 7, 8 groups, and one group survived. We harvested about around like 200 babies, like spats. But unfortunately, that's 200 oysters lost during a storm in that fall. In 2019, we tried again, and actually we did got like unfortunately the larval survival is really low, and we tried to tried to culture this stuff in the lab, and maybe thinking to give them a good condition, but we only have several oysters survived to the adult stage. Right now it's just 2 or 3, so it may not be able to build up any like a founder population. That's basically this is the tetraploid product, and I have been working in the lab. Hopefully in the future, if the current pending proposal can be funded, or even not funded, if the industry needs some like extension way for help to do like whatever, think like I could be helpful, that definitely I would love to help with this topic. Next, please. Okay, another one, researcher in my lab in the oyster, like about the oyster, is a germ plasma cryopreservation research. And basically, for this part of research, we are trying to streamline them and improve the existing protocol, which I worked on in 2012, published on the aquaculture. Also, we wanted to see if before that, we all know, and Gulf oyster industry aquaculture is growing rapidly. We will see if we'll be able to establish a sperm bank for the Gulf wild oyster resources. And another thing is maybe later you will hear Kelly will talk about the breeding program in the Gulf, and be able to participate with that product to help with the archive of the males and from each generation. And of course, there's another use for this technology, we preserve the strain or lines if there's created a line, created a strain, we'd be able to do that for sure. And also for the tetraploid sperm. And it's also within my reader. Hopefully, we will see how that goes. Next, please. So that's the two papers which I mentioned. The first one is in 2012 I published, and we did in the another way we did research about using the cryopreserved sperm from the oyster, and then when second year when the oyster turned into from a male turned into female, we be able to do the self-fertilization to create a highly inbred lines, which be a very speed up way for genetic breeding. Next, please. Basically, so for the Gulf oyster resources, and trying to, I wanted to combining, this is kind of combining the breeding program. We want to see if we be able to get the oysters from each different locations. And, I mean the wild oysters, It's not like just want to preserve these genetic resources in case in the future we need them. We will collect their genetic data or biological data, preserve them, and post soil motility and evaluation, and then inventory and the record. Hopefully, this part of work will be linked with the USDA and the genetic bank, and for future use, just like the livestock. Next, please. I think that's all I have to talk about the oyster work in my lab.
Okay, thank you, Huiping.
Thank you.
That is, yeah, absolutely. Thank you. And then Scott Reichert from Auburn University Shellfish Lab is up next. So Scott, you got to take yourself off mute, but I've got your slides here.
Okay, I should be off mute. Okay, just a quick update from the Auburn University Shellfish Laboratory. We've been working on a Florida line of tetraploids for several years now. The original stock was sourced from Apalachicola Bay and Cedar Key. We made a chemical-induced triploid, raised that for a year, and then made a chemical-induced tetraploid. Had real good success with that. And actually today we are doing a new generation of that Florida tetraploid line. We hope to have a very large pool of that source available going into next year. We do have some of the Florida tetraploids that were produced last year that we, now that we have done this spawn today, we're going to start allowing distribution of some of the Florida tetraploids going forward through this summer. We are also in early development of a new tetraploid line from one of our selected lines. We call it our Albee line. It's a line we've been working on for a few years now that has been selected for shell shape and cup depth. So it's pretty much Alabama genetics, but it will be a new tetraploid line that we hope will be ready for distribution in 2022. We've made the chemically induced triploid line for that, and we will be producing the tetraploid line from that this summer. So that will give us 2 new lines going forward. Also, each year we produce a new generation of the existing Louisiana tetraploid line for distribution. And what we've done to date is use the newest generation to produce the next generation. What we're going to start doing now is actually going back several generations and using older animals that have been on the farm for several years, and take those animals that have survived quite a while and start producing our new generations from some of the older stock instead of from the newest generation that might not have faced a lot of adversity. So hopefully that will produce a little bit more resilience in that tetraploid line. Next slide, Bill. And lastly, I just want to mention that we do provide services at the Auburn Shellfish Lab, working with hatcheries to provide tetraploid sperm for production of triploids. We do this in conjunction with 4Cs. So we provide tetraploid broodstock, tetraploid sperm, and then we also can provide those hatcheries ploidy verification of their seed and certification of those seed as triploid. That's it for me.
All right, thank you, Scott. Appreciate it. And that brings us to Dr. Kelly Lucas over in at the GCRL's, the Thad Cochran Marine Aquaculture Center. I think I got that right, Kelly. So I should have your first slide up there. I'll let you talk about salt.
All righty. So SALT, or the Selection of Aquaculture Lines with Improved Traits, is a Gulf of Mexico genetics and breeding research consortium. And this consortium I have listed there, the established in 2019, if you would. This was our first round of funding in 2019. A lot of you on the call have been involved in discussions to get a consortium off the ground for the Gulf of Mexico to look at breeding oysters. And I know we really seriously started those efforts and talks back in 2017. And so happy to have that off the ground in 2019, but I think it just kind of also shows, you know, not only are you dealing with biological timescales, but also these funding cycles and what we can accomplish based on when funding comes in. As you see there, the research partners listed. So participation across the entire Gulf, USM, Auburn, University of Florida, TAMU Corpus Christi, 4Cs, Tom Rossi's on there, as well as Louisiana Sea Grant, right? But we also have a business advisory council. We really helped form this consortium with a lot of industry participation. And so we have a business advisory council that helps guide what we're doing at the consortium. And as you can see, we also have representation from each of the states there. And several of those who were on the advisory council were kind of there in 2017 when we started having these discussions regarding salt. Bill, you can go ahead and flip the slide. So really our purpose is to assist industry and as well as our state agencies by developing these genetic resources for eastern oysters of the Gulf of Mexico, right? And creating a breeding program to improve production and market value traits as directed by industry needs. Bill, go ahead and flip. So the first thing we did that you can see on the right, as I provide some of these highlights, is really to get a sense from industry, what are your desired traits that you're looking at, right? What is going to help you? What do you need? And so several of you should have received, everybody, we tried to compile it as comprehensive as we could, a copy of these, the survey to fill out. And we sent that back in late January, early February, and did some follow-up in February as well. We are just now compiling all the results from the survey. So if those of you on the call would like to respond, we can still slip you in, but I can tell you just kind of overwhelmingly, just as your concerns on this call are, it's all mainly environmental related concerns that you have, whether it's dealing with variable salinity or specifically high salinity or high temperatures. Most of your concerns are geared towards that. And so, you know, we as a consortium research partners, we hear you, right? And so this is what we want to work on. You know, if we're to pull broodstock and pull oysters from around all the conditions of the Gulf of Mexico, you know, can we be more optimized? Can we optimize these oysters for the conditions in the Gulf? is what we're looking at. So the funding was initially awarded in 2019. It's 5 years and it's contingent on federal appropriations to the program, right? So we receive funding in yearly cycles, even though it's already been established that this is a 5-year consortium. So we have 2019 and 2020 money is already in place as well. So some of the year 1 milestones here are listed for you. The survey of industry needs, so we can work on those breeding objectives. We've been collecting broodstock. Your representatives that you saw there have been across their states collecting the broodstock that we're going to use. Those broodstock are maturing in location. We're looking at the genotyping and cryopreservation that will occur this year, as well as our first round of spawning, which has been just slightly delayed because of COVID. And so it will occur in late summer, early fall, if you would, just based on the delays associated with our current pandemic. But that's just a overall high level of the SALT program, and we look forward to continuing to work with industry to meet these challenges.
All right, thank you, Kelly, appreciate it. Let's see, Brian. Kelly, if, by the way, if you have a chance, I don't know if there's a link to that survey that we can put in the chat or not, but while I do that, I'm going to pull up Brian's slide. Brian, you should be able to see your slide here, and I may have to click it forward, I think, right?
Yeah. So briefly, just to cover some of the work that we're doing in Louisiana that hopefully directly addresses these mortality issues, it can be broken down to continuing to investigate the root causes, which is all of the things that we heard about in the first part of this webinar. What do we know about them and what don't we know, and investigating those types of things. And we're doing that mostly through collaborations within the state and across the region to try to find out what common threads are people experiencing, what do we see, what can we detect. And those types of investigations include looking at the genetics. We heard some great information that earlier, talking about how, what broodstock you use to produce the seed matters, how you handle your oysters might matter, the types of gear you're using might matter, and then the interaction of all these things with the environment, with the gear, with the specific genetics. So we're going to continue looking at those things, testing the combinations, seeing what works, what doesn't work, and when we see what doesn't work, hopefully that'll continue to inform us of what maybe is causing some of these things. And the other component really is breeding, carrying on with the breeding efforts that have been going on at LSU for a long time and across the region. So, there's been a lot of talk about tetraploids. There's been Tom and Weeping and Scott have all talked about new tetraploids. We're also working on new tetraploids, trying to get as much genetic diversity as we can, and then hopefully that will carry over into the triploids, and maybe we can start improving the tetraploids to have better triploid survival. And also not just survival, but other important traits too. So tetraploids aside, we're also looking at diploid oysters. We're testing currently a number of different populations that we've taken from across Louisiana specifically for us in a wide range of environmental conditions, from really fresh water areas to some really salty waters that have a lot of dermo disease presence typically. And with hopes that, one, we can make better triploids with those, but also there's been a lot of interest recently in using more diploids on farms. So we want to make sure that the diploids that are available are the best that they can be. So that if you're buying a bunch of diploid seed, it's going to perform as well as your triploids, and it's a really viable alternative crop that you can have on your farm. And yeah, so like I said, all of these is really collaborations throughout the entire region, because everybody's experiencing similar things in a big variety of environments. So it's really important that we continue to work together, both as researchers and with the farmers, to ask these questions and find the answers as soon as we can. And that's it.
All right, thank you, Brian, appreciate it. And then we've got Dr. Stan Allen. from the Virginia Institute of Marine Sciences, who's going to talk, give the perspective from the East Coast.
Yes, hello everyone. So what I'd just like to do is very briefly describe the approach we've decided to adopt, because we've been making tetraploids since the early 2000s, and we've been doing diploid breeding for even longer than that. So we have been asking this question about what to do about triploid mortality recently, since it really arrived in about 2014. So we've gone through some evolution on what we think about things. And so we're all familiar generally that this triploid mortality issue or unexplained mortality issue varies with the environment. Of course, there's not much we can do about that. So we're looking for genetic signals about this problem. And our initial work, and it's also ongoing in the Gulf Coast, is to explore a new variety of tetraploid that may add to, may improve the situation somewhat. The Florida tetraploid, the Gulf tetraploid. We have a couple of different tetraploid lines ourselves that Joey talked to you about, and we tested out. And generally speaking, it doesn't seem to make a big difference who the tetraploid was. But let me just emphasize that we're in very early days on that, so we can't rule out that perhaps the Florida line will do a much better job for the Gulf Coast and that kind of thing. But to make progress in a systematic way, we really want to have a much better idea of what makes a difference in the triploids as a function of what tetraploids went into it. So there's been a couple bodies of work recently. The first by our grad student Kate Ritter, who made some triploid families from individual male tetraploids, and she was doing a different project, but at the site where triploid mortality happened, there was a very large variance, in other words, from good to bad, among those families in whether or not they exhibited triploid mortality. So that was telling us that the tetraploid parent, the male, made a difference. More recent work by Joey in his dissertation, where he's going much deeper into this subject, has shown the same kind of thing. The tetraploid male matters. So this is the type of information that suggests to us that breeding could help. So next slide, please. So our initial approach, as I've said, we've had a diploid breeding program for a very long time, developing lines, and also a much more precision approach by developing families. And that has led to improved diploids, which obviously is the point. But, you know, we also started doing that with tetraploids, primarily because the tetraploids were misbehaving. They weren't very good broodstock animals, and we wanted to try to domesticate them. So we started to do a parallel program with tetraploids, developing lines and also a more precision approach using families to get improved tetraploids. And initially, we surmised that if we improve the diploid and if we improve the tetraploid and we cross them together, we ought to get an improved triploid. And I must add that, you know, the diploids were being chosen for the usual suspects, like, you know, growth rate and shape and survival, and so were the tetraploids. So we thought moving both of these along in parallel will provide us with an improved triploid. Next slide. But the problem is that triploid mortality isn't a usual suspect. In other words, the triploid mortality happens in triploids. It doesn't happen in diploids, and it doesn't happen in tetraploids because they're not triploid. So it doesn't immediately follow that if we improve diploid breeding, in diploid survival, and we improve tetraploid survival, that cross will necessarily yield a improved survival triploid, because there's a different mechanism driving these triploids than is driving the reproducing diploid and the reproducing tetraploid. So this really is a bit of a conundrum about what to do. So our approach now is illustrated on my last slide, which is basically continuing our diploid breeding program and our tetraploid program such that we have pedigreed families and we know what to expect from them, and using those pedigreed families to make pedigreed triploid families. And it's in those triploid families that we can measure survival in the triploids. Again, diploid and tetraploid survival doesn't necessarily indicate triploid survival. So we pick out the best triploid families as a result of this test. And obviously we can't breed triploids because that's why you want them, they're sterile. So we have to go back to which are the best 2N families that gave us good triploid families, and which are the best 4N families. And this is now our approach. And this is the relatively new science that Joey Matt is starting to develop with his PhD work. And it's basically where we're at right now, making, unfortunately, 3 sets of families, diploid, tetraploid, and triploid, because of this damn triploid mortality thing that can't be measured in tetraploids or diploids. Thanks, Bill.
All right, thank you, Stan, appreciate it. All right, and we've got one last talk. Let me pull this up. Let's see. Of course, finding this will be the hard part. Let's see. There we go. All right.
I guess I'll lead off on this one, Bill.
Yep. Yeah, please do. There you go. Should now be sharing.
All right, great. So Bill and I would like to introduce a collaborative and regional approach to applied research that brings together not only researchers, but extension specialists, local agents with our commercial growers. It's not unlike the approach taken in some of the projects that you heard about earlier on today, or some research projects completed recently in which you may have been involved in. But we're giving it a name, oyster farms. Farms for oyster aquaculture research and monitoring of shellfish in the Gulf of Mexico, and suggests that, that this provides a framework for any and all experimental field work. So here's a few upcoming oyster farms projects.
So I'll start with this one then. So the obvious one is doing a PLOIDY comparison. And so in particular, and I understand what Stan just said, that the tetraploid line may not be as important as we'd like, but we need to test that. And so the first thing that we'd like to do that's underway right now is comparing triploid seed that are produced from the traditional Louisiana-based tetraploids with the Florida Gulf Coast, West Coast tetraploids, and comparing those. And they would all be compared with half-sibling diploids. We'd like to deploy those in at least Mississippi, Alabama, and Florida at commercial farms that could represent the different environments, and do that, ideally plant those this summer and follow them through next year. We would not be able to, because of some of the seed restrictions, we can't take half-sibling diploids and get those to Florida. So we can't do that comparison, but in Florida, but we would be able to do that in Mississippi and Alabama. I would just emphasize it's not a large amount of seed. We can't do that, but it is enough seed that we try to get some results to see if we do see some differences in those different lines. I'll go on. The next one here is Leslie.
Okay, we just received just a small bit of funds in-house from the University of Florida. That's going to allow us to look at some monitoring of our farms, particularly in Alligator Harbor and Oyster Bay. The intent is to assess the relationships between water quality through continuous monitoring, phytoplankton abundance and composition through weekly monitoring, and health through bi-monthly monitoring, and compare that with oyster survival. We intend to provide some of the triploid oysters spawned from now Florida West Coast specific tetraploids to growers in September, a fairly large seed that would be nursed initially. And then run that through June of the following year so that as the oysters are reaching market size, they're being exposed to that spring, early summer mortality event. So we look forward to working with a few growers there and providing that information later.
Important that I'm not muted when I'm talking, if I want to communicate at least. The last part of this is the SALT project that Dr. Kelly Lucas mentioned, getting these improved traits. And so the idea here is instead of selecting survivors from particular environments and doing a breeding program, the idea here is that you want genetic diversity. And so this year we are creating what's called a mosaic line, and that's essentially trying to get genetics from Gulf of Mexico oysters mixed in the hatchery, make a giant, reshuffle the genetic deck. And so then take the offspring that are generated from that reshuffling and plant them out at different farms in different conditions. We know salinity is a driving force in the Gulf of Mexico. So plant them out under different conditions and see how they perform. Obviously, you need good survival, but you also need good growth. And ideally, you've got an oyster that doesn't turn into a shoehorn shape either. So those are the types of things that we plan to be looking at. The selection beyond sort of the basics of survival and growth, some of the selection traits can be driven by that industry survey that Kelly shared earlier. So our intent is to get that out. The seed from that spawn should be large enough that they'll be out in farms, planted out in the early spring, in which case they would be exposed to whatever this spring / summer / warm temperature mortality events are. And we've got some sites that we'd like to have commercial farms in Florida, Alabama, Mississippi, and Louisiana where we can do that. And that is all part of the consortium that Kelly is leading up, but you can see that there are others involved with that. That may be, yeah, that is our last slide. So I think, Rusty, I think it's off to you there.
Yeah, Bill, you're going to throw the slide up for the survey as well. So yeah, sure. Thanks to our presenters from the second round. We'll go ahead and take some time here for questions. And again, just to remind you, if you'll type those into the chat, then Natalie will read those off for us.
So I have a few questions for Tom from 4Cs. For the hatcheries that wanted the new tetraploid line from BROODSTOCK, were they given this new line or did they have to purchase the new line?
Okay. That's for me, right?
Yes, Tom.
Yeah, so I can answer that one. So we actually distribute the tetraploid stocks to our partner hatcheries without any charges. Right now, each of the hatcheries in the Gulf that'll service Florida have a couple hundred adult tetraploids ready for use, and they have access to as many as we may have. And there's multiple thousands available at this point. So there's no upfront charge. However, we do require the hatcheries to charge a fee for the use of the tetraploids based upon success. In other words, we don't make the hatcheries pay anything up front. We ask them to charge a fee to their customers, you, the grower, if you would, And the fee then is passed back to us, distributed to us and our partners to fund and finance the development of additional tetraploids in the program in general. That answer it?
I think so. I also have another question for you, Tom. Are you guys developing a new tetraploid line for only the Gulf, or is the east coast of Florida lines being developed too?
Right. So the answer is West Coast in Florida or Gulf only. I can tell you that I have not seen any real need or call for a triploid oysters on the East Coast. So if there's no need for triploids, there's no need for us to develop a tetraploid parent to service that. And in Florida, you have very strict rules. We can't, well, might be some exception for tetraploids, but generally you can't cross from one side of the state to the next with seed stock. So the answer then is at this point, no East Coast tetraploid development ongoing.
Tom, has there been any progress in other states though on the East Coast?
Oh yeah, we have tetraploids in, well, going up the state. There's Georgia, well, we don't have tetraploids there. Georgia has a very experimental, small oyster grower population. We're working with the University of Georgia to produce triploids, but they use tetraploid broodstock from the Gulf, basically. South Carolina does have existent South Carolina produced tetraploids, and North Carolina, no. North of North Carolina, those hatcheries are serviced by the Virginia Institute of Marine Science, or STAN's operation at ABC, and he takes responsibility for growing the tetraploids for use from the balance of the states north of North Carolina.
I just want to interrupt and clarify that according to Florida's rules or BMPs, that triploid stocks developed from Gulf of Mexico tetraploids can indeed be reared in the east coast of Florida. So you do have that allowance.
I have a few questions for Dr. Hoi-Ping Young. Is there a sperm bank for East Coast wild resources for breeding programs as well?
I think that's I answered that in the chat to everyone, and that, of course, that would be great, right?
Okay, so I have another question for you as well, Hoi-Ping. It says, what QC measurements besides motility and post-thaw motility will you use to streamline oyster cryopreservation?
There's for that the post-thaw quality, of course, the fertilization would be the golden. Fertilization is always the number one, but considering sometimes the female is not available, we also use post-thaw motility. Also, for the use the flow cytometry in the lab to do some analyze the mitochondria of the sperm, which is kind of like a motor to make the sperm moving. Also for the cell membrane integrity. That's the three things that we measured in the lab. And trying to, this kind of measurement could be correlated with the fertility. Actually, I can say that based on, for example, for the oyster, the published paper from me 2012, the fertilization of crop preserved sperm is about diploids. It's 50 to 55%. That's the fertilization.
I have one comment. It's actually from John Supan, and I thought it was really valuable to share with the group. He's with Navy Cove Oyster Company, and he said at Fort Morgan, Alabama, there was triploid mortality hotspot in 2016. At that time, the cause was attributed to compound stressors, particularly during the freshet. Since then, they haven't experienced high mortality, particularly during this year when they made it through a long 4-month freshet. They implemented 2 particular farm management practices since 2016 that seem to be fruitful for them. One, they reduced compound stress. They did no aerial drying during low salinity. If you can taste salt, don't flip. Number 2, they changed to 25mm mesh bags for final 2 months of grow out. This dramatically reduced matting of microalgae on the surface, which can trap silt and cause muddy conditions within the mesh. This also greatly reduced the occurrence of mudworm blisters in the harvested oysters. So I just wanted to say thank you again for John Supan sharing that.
I would just add to that that was one of the sites where Pandora did her study in 2017, where we weren't handling those oysters at all. They were side by side growing at the site, but we were not implementing any management procedures like Soup described there. They were just in the water. And we did see a significant difference by the end of the year where the diploids had died at a higher rate. I'd have to go back and look to see what those percentages were, but that probably adds to Soup's point that when he managed it, they felt that they had less mortality going on.
Any other questions, Natalie?
I'm not seeing any other questions in the chat box. I have provided everyone's contact information for Bill Walton, Leslie Sturmer, and Brian Callum. That's a mouthful. There's also the survey as well, if you guys could complete that to evaluate the workshop.
Very good. Bill?
All right, we'll just give a minute here. Does anybody else that got online? I appreciate people doing that, but does anybody else have any comments? It doesn't have to be a question. You're allowed to make comments. In fact, I think a question just showed up. I've got it. Yeah, I've got it, Natalie, right here. So our oxygen levels, I think that's I'm not sure how to say that name, MEFD. It's probably not, sounds like a gang name the way it showed up, but our oxygen levels monitored, and I don't mean any offense there, by the way, our oxygen levels monitored inside growing cages during these mortality events. That's actually very interesting. That's one of the things that came out of some of the discussions with growers, is it's been frustrating that we can have monitors in the water, and anybody, Jerome, Joey, speak up if this is different for you, but or confirm if it's the same. We've put different sensors in the water for salinity, oxygen, temperature, and there are times when we're at a site where none of the levels look to us like they should be causing mortality, but we see mortality. And in particular, I think the point that was made, Leslie, you mentioned this earlier, and maybe Ryan confirmed this too, that some of the bags that were being talked about out of the panhandle, people were talking about these amphipods and anoxic mud in the bags. So the sensors that were in the water there said that the water conditions were what you would call good for oysters. But it does make you wonder if the growing conditions in the bags are not. And there's a couple ways you could get to that, obviously, and fouling is one way that could happen. You know, you've all seen bags that get fouled up, and you could imagine that conditions in those bags could be very different than what's happening outside those bags. So that is one of the things I think that we, that's our intent, is to look at that going forward, because it's hard to get sensors inside those bags. And so that's not how the typical, YSI meter works. But there are probably ways to do that. Leslie, do you want to chime in?
Well, we certainly need to explore that. I mean, that is the data loggers we intend to purchase with that monitoring project will not include dissolved oxygen. Not even sure. We're going to look into it. Let's just put it that way.
And Bill, I'll mention, I can confirm what you said, for one of the research projects in the Chesapeake, we had a sensor monitoring dissolved oxygen, among other things. It wasn't in the bag. And so, yeah, I could see there could be some variation there. But before, during, and even after the mortality event, the dissolved oxygen levels were high. I mean, they were not at a level that would be considered stressful. And now I've actually switched over for more easy to measure things like temperature. of putting monitors in the bags as opposed to attaching them, you know, outside of the bags or whatever. But I would also say for things like salinity loggers, we've had struggle with those being reliable. So if you can do it, I would put 2 salinity loggers at each site, so you can confirm that they're getting accurate data, and in case one fails for a certain time period, which they tend to do.
That's a good idea, Joe. And that monitoring program we hope to implement, what we plan to implement in September, that is the idea. We'll have 2 farmers at each of the lease areas. Actually, tomorrow we'll be sending out some of the small hobo temperature data loggers that continuously record about every 30 minutes. We have some growers lined up in the Panhandle, and the intent is to put them inside the bag, so it's monitoring temperature through not only the culture period, but when they're doing their various biofouling, control, tumbling activities, just to get an idea of the temperature that goes on, particularly over the summer months. So we'll be sending those out to a few select growers that have volunteered to do that this week.
And Jeff Clements commented, by the way, that the noxic mud is interesting from a bacterial perspective, because even low O2, low oxygen, can have bacteria proliferate, and that's what can kill the oysters, not necessarily the low oxygen. So maybe that may be a productive route to go. While right there, Doug has asked, does the growth on the bags, the fouling, does that consume food and oxygen levels that oysters are trying to survive on? And you know, I'll say it depends on the fouling. There are certain fouling organisms that would directly compete for food. Certainly wild oysters on top of your bags will do that. Others would not. But even if they're not directly competing for food, if anybody has, like in Florida, we've seen some of the soft fowlers, like some of the tunicates, the Ascidians, like they'll start to, or those amphipods, those tube builders, will actually block the mesh. And so even if they aren't eating the same food that the oysters are, they've effectively made your mesh bag a solid bag.
And they're respiring.
Oh, good point. Thank you. Excellent. Yes, that's a good point. There's more than food that might be limiting. There's oxygen. So yes, they could be using oxygen. And then you could get decomposition when a bloom of those things dies too. Yep. So, thank you. All right. All right. I think at that point, Rusty, I think we've answered the question. So I think we'll say, any of the panelists have anything they want to add?
Well, I would like to do a shout out. I don't know if that was in your wrap up talk, but certainly you are going to recognize both Rusty Grice and Natalie Simon here that have done the lion's share of managing the technology here. But actually the biggest shout out goes to you, Bill, as always, being very responsive to industry's needs. And really this workshop, virtual workshop, is really in thanks to your efforts. I'm very appreciative, and I'm sure all the growers that are participating as well in this virtual workshop feel the same.
Well, I appreciate that, but it is a team effort for sure. And I thank Rusty and Natalie and Leslie. Thank you for everyone that attended as well. I know it's a big chunk of your time. I hope it was worthwhile, and we appreciate your patience. Thank you to the panelists. It was a big chunk of your time as well. Appreciate you making yourselves available. We do have that short anonymous survey. Natalie or Rusty have put that in the chat. We would love to get your feedback, because yeah, that is a place where if you have observations about what you have seen on your farm, patterns that you have seen about mortality, if you have suggestions for what we should be researching, observations, that is a great place to do that. Please do that. You've got our contact information here. I think some of our panelists may have shared their contact information in their slides. If they didn't, and you want to contact, for example, Jerome to follow up about some of that work that he did, or any of our panelists, please contact us and we can put you in touch. Again, we hope the workshop's useful. We do want to work with the industry to help with this critical problem. We understand that this is your livelihood, and our intention is to try to help with these issues. So with that, we will make the recording available online. We've got a link for that as well. And I will say thank you everyone for coming out.